For the second speakers, he is a senior lecturer from UC Malaysia, Terengganu, UMT. He's obtained his PhD from University of East Anglia, Norwich, UK. His expertise in research is the exploration of nanotechnology on cosmetic and pharmaceuticals, which one of the backbone to the development of biotechnology in Malaysia. He is also actively collaborating with Unilever Sdn Bhd, which have interested in collaborating to develop nanos vesicles formulation in their product. And he also was a former director of the Management Center RMC, University Malaysia Terengganu, and currently actively commercialize his research product as well as working with several leading companies, including Rafalina Sdn Bhd and Halal Gel Sdn Bhd. For your information, he have published his work in ISI, WOS corpus with more than 45 journals, patents, copyright, trade secret, and trademark. He has secured national and international grant and also received 20 research grant, more than 2 million, and become the PI for the 12th part of the 20 research grant. He has supervised and co-supervised around 50 students, including 5 PhD, 5 master, and the rest are undergraduate student and co-supervision. He received many awards such as gold and silver in national and international, such as SciTech Innovasi UMT, iTech MTE, the national and international such as Seoul Innovation and Invention Fair, and also produce product BF Nano V Emerging, the secret of rapid skin rejuvenation, and thermo gel for smart muscular system in the My Innovation 2020 competition. He received a very great achievement, has the best performing team in the business plan in capacity building for innovation and university R&D commercialization by Stephen Institute of Technology USA and UKM. Without any further, I would like to welcome you as our speaker, Associate Prof. Chem Dr.Mohd Zul Helmi Rozaini. Please, Dr. Zul, share your slide.
Thank you very much. Can you listen to my voice?
Yes, clear, okay. Dr. Zul.
All right. Thank you very much. Thank you. Sorry. Thank you, Mr. Chairperson, for the brief introductions. And thank you, Dr. Nick, for the detailed explanations about the challenges and strategies in innovating biomaterials. And I think this will be the case study on how we apply the usage of biomaterials in cosmetics. Okay, and ladies and gentlemen, and I would like to share on how we use the natural resources in coastal area for us to transform into materials that have I think a greater impact on human and also communities. So first of all, I give you the scenario of the problems here in Terengganu or in Kuala Terengganu or Kuala Nerus, the UV rays, okay, because we are in the coastal areas, either from the sun or from the artificial sources like tanning beds. It can cause sunburns, and exposure to UV rays can cause premature aging and to the skin, and the sign of sun damage such as wrinkles, leathery skin, liver spot, actinic keratosis, and UV rays also can cause an eye irritation, eye problem as well. Okay, why we use sunscreen? And besides protecting you from the sunburn, why should you wear sunscreen? And here's my top 2 reason actually. The sunscreen prevent premature aging, and UV radiation can cause a premature breakdown of collagen and elastin in the skin, okay, and resulting in the very fine lines and wrinkles. Okay, and sunscreen also helps to prevent sunburns and I think skin cancers. The most biomass in Malaysia is oil palm. Okay, fat. Only 10% of the oil and all 90% is biomass. So any research related to oil palm, it must be secure a research grant here in Malaysia because they want to solve the issues of the oil palm. And until now, almost all part of oil palm can be used. But in Terengganu, the problem is different. Okay, it is not as bad as the other side of Semenanjung, Malaysia, or other states, but because of the main industry here in the coastal areas community is the crackers, okay, keropok, right, keropok manufacturing, and the aspect of waste utilization is the main concern because only 45% of us use fish meat for industry, and another 55% is the waste. So we throw away the fish bones, fish stomach, and fish head. So imagine what it would be like in 20 years' time, ladies and gentlemen. So why we use the fish bone, okay, as the subject? It's because it is abundant of. I think Dr. also agree with me. We have a lot of hydroxyapatite in the bone, in the fish bone, as a main component, and the presence of phosphate ester group and carboxylate group at the collagen surface, and also the apatite as the extracellular fluids are supersaturated in calcium and phosphate, and also being a good protector from UV rays and can be a metal doping as well. Of course, the bone is non-toxic. That's why it is very biocompatible. Okay. So sound cliche, although the concept is already cliche for us. Turn waste into wealth, but I think it works. Okay. Before this, if you want to get the bones, can get it for free. But now, if you want to get the fish bone, we have to pay, right? So they can generate the income from it. So, ladies and gentlemen, I divide this presentation into three phases. Namely, the first one is on the development phase. The second one is the prototype phase. And the third one is commercialization phase. Okay? So, can we look at the timeline, which is the problem? What is the problem that you are trying to solve? This is the fundamental part, okay? The development part, we always hear about the effects of UV radiation on the skin. And for the coastal environment such in here, it gives a serious impact, especially to the fishermen, because it's the main activity. They are exposed on the UV rays, and this is vulnerable for them, okay? So the issue is on the UV radiation. And on 2017, we awarded the FRJS, okay? And what is the result of this grant? First, we able to get the information about the 41% of the percentage yield. We can get the hydroxyapatite for 100 gram of fish bones. And the second one is the formulation or the process producing the matrices from the phase diagram formulation. We name it as nanovesicles. And the third one, we already filed the copyright, okay, under the my IPO. So next, I would like to share the flow of works. Okay, so the first we get the fish from the factory, or sometimes we get it from the fisherman. Okay, and we clean the fish, all right, and then we have to cook. Have to clean it first, and after all, there is a machine that separates the bones from the meat. And last but not least, we go through all the process such as calcination, and at the end, we able to get fine fish bones. After that, we have to grind it, and we have to extract using the water distillation, steam distillation process. Okay. This is the first part of the development process to synthesize the modified hydroxyapatite or fish shield later on. Okay. So now I mentioned what is the process like, and after that we have to characterize the hydroxyapatite using NMR, XRD, FTR, and ChM. We do it a bit on the rheological stable and stability, and also the SC and thermal transitions. Okay, and this is the physical appearance. You can see now the pure fish bone is in the white colors, and the modified one is in brownish and reddish colors. Okay, so second one, it goes straight to emulsion formulation. Okay, so this is called the phase diagram. Okay, the tri-phase diagram that measures the complex possible interactions. During the formation of unique phase resulted the mixture of several components. Each edge of the triangular diagram represent one component and measured in weight percentage of the emulsion in the system with increasing concentration. And in cosmetics, the ternary phase diagram is constructed to determine the mixing ratios needed to prepare a stable microemulsions. Okay, so we can construct all the phase diagram, and later on we're able to do the pH studies and also the stabilities, and at the end we can choose the most suitable phase diagram and come up with all the formulations. So we have all the formulation as one. The next goes to the prototype phase. Okay. If we, when we want to move on to the prototype phase, we need to identify which TRL is the product we want to bring. Just now, Dr. Nick have mentioned earlier. All right. So what are aspects that we need to obtain to commercialize the products? Okay. So now we are looking for TRL 4 to 6. Okay. And if you look at the timeline, okay, the second phase is about the prototype phase, which is on April 2018. We managed to obtain the UMT commercialization grant amounting about 20,000 as well to conduct several validation tests, such as eye irritation test, shelf test, heavy metal test, and cytotoxicity test from serum. And we finally have registered the product name under name Zaura. Okay, we got the NOT numbers as well here. And then from the NPRA. So we also already worked out to get the PRGS grant on 2019. And yes, at the end, this prototype grant is able for us to commercialize the current products. Later on, I will show you. The main challenge is to place a fish shield, or later on that I mentioned the modified hydroxyapatite, as a UV filters on Annex 7 of the NPRA guidelines. Okay, so we have Annex 7 Part 1, we have Annex 7, we have Annex 6. So this is the list of filter that we have to announce, or we have to determine. Later on, the fish shield should be here. Okay, that's what we aim to, right? So this is the guideline that prepared for the NPRA for us to come out with the products. Next, in the PRGS, in the prototype phase, we also have to validate the SPF values. Okay, if I mentioned before this, our SPF is 50, so we have to validate it. Okay, so we're using the sunscreen analysis in vitro and in vivo techniques, and all of these techniques we gave it to serum to determine, and also we send it to Bangkok for further verifications. Okay, so far we're using Manso equation to detect the solar intensity or the solar spectrum by Sari et al. Okay, so yes, at the end we managed to get the SPF 45, nearly 50, and we do some a bit media works well as well. So come in the harian, and then just want to review about the potential of the product that can be commercialized. Okay. Next, we go to the commercialization phase. In the commercialization phase, we have to move on from the TRL 6 to TRL 8. This is where we need to explore about the business plan and how to involve with the industry player. Okay, we have all the ingredients. Okay, we have all the stuff. So we need to identify which industry, or are we going to come up with our own business entity, or like me, I just do the joint venture with UMT Jaya Holdings. Okay, so try to have a look. This is the commercialization phase. Okay, so we are successfully paired with a new company called ATEX PLT under the Symbiosis program by MTDC. Same with Dr. Nick mentioned just now. And we finally also managed to promote and collaborate with the leading local companies such as Rafelina that based in Kedah to jointly market our Zaura as the latest cosmetic product. So Zaura basically in Arabic means shield or yeah, shield. That's why we name our product because we are doing something like sunscreen. So we call it as Zaura. And we market it as a premium under Sisfel or Rafalina for the cosmetic for women care brand with a glorious launch in 2020. So it has been launched on July last year. So how is protected? So actually, the raw material, we produce the fish shield, I mean the modified hydroxyapatite by our own and leave the extract in our own laboratory. And then we send it the 20 kilograms to the fish shields to the OEM. Okay, so they set up until 10,000 Zaura DD cushions, and that's why here we really need the intervention from certain parties if we are interested in collaborating further. Okay, so for this product, we already have the whole IPs, patent, we have the copyright, and we have the trademark as well. Okay, so that's why I think really kind of protected. So I would like to share a bit of promotions that we've done. So if anyone is interested, you also can call Dr. Fatia, me, or Dr. Nick. Oh, sorry. Love, thought this was there. Aha. Yes, this is our solution. This is my product actually. We call it as the Zaura DD cushion, cream cushion. What is DD? DD means daily defense. Okay, so what is so different with other DD cushion? I think uh, the- we have to come up something different. Okay, why not we try to combine three different items in one cosmetics, one systems. Okay, we have a foundation, we have a moisturizer, and we have a sunblock. Okay, if you can have like three different products in your bag. Why not you just come up with one? You can sum up with the all perfect one products. Already the cushions. Okay, I'm promoting my product as Dr. Heavy. It's okay. So, and then we look at our competitors, okay? Lineage, we have a Dior, and we have a Zafisha foundation untuk mak-mak je. Okay? So that's just how we want to compete. All right? And this is the business model that we through. Okay, UMTJ, because my product is under UMTJ Holdings, the business entity under University Malaysia Terengganu. We produce in the OEM, okay, and then we give it to Rafalina Serembahat to market it through the Sisfel Empire. I don't think so. We have in Shopee at the moment like Dr. Nick, because we have a lots of agents, about 6,000 agents. So if you want to buy it, and then you have to go through the agent. So how it works. Okay, and this is the distributor, the Rafalina factory. Okay, it's in Kedah. Yeah, this is actually I don't know if you know or not. Before this, Rafalina already sponsored the Astro Ceria, or we call it Astro apa benda Astro rancangan kanak-kanak. I don't remember. Okay, like Dr. Vida sponsored the Gegar Berganza, right? Dr. Alif also sponsored the Gegar Berganza, and Rafalina also sponsored because they have a child product. Yeah, this one, the Caliph. Okay, mama nak kali. So that's why I say we have to identify the industrial that can move us forward. Okay, so they will marketing my products as their product as well, and we will share the profits together. Okay, so this is a bit profits. The 85 until it's only 85,000. I think it's okay. In 6 month time, we got 85,000, right? In the COVID pandemic keadaan sekarang kan. So this is the CEO and also the co-CEO, his wife. Okay, so yeah, and we come up with the three what called three tone. We have Elmer, natural, and porcelain. It's like K drama or K model, right? So this is something that we always playing about. Okay, we need all the recommendation by the consumers, and we put it and we can market it, right? So this is the event with all the agents, and I think that's all. This is my part. We have to promote it during the Lilah Barangan Buatan Malaysia, okay, and also this was the brand of Buatan Malaysia, and then yeah, they appointed me as one of the advisors. Okay, so we have Sheikh Khalil, we have Ibu Hanim here, and me. Wow, all right, and that's all. Thank you very much. Thank you, Dr. Fatia. Back to you.
Thank you very much, Dr. Zul. Very, very wow presentation from you. All right. I think both speakers have presented a very informative, interesting, tremendous presentation today. And now I would like to open for the question and answer session from the floor, from the participant. Any things that you would like to ask from both our awesome speakers? You can unmute your mic,
Okay.
can ask directly, or you can write in the chat box. All right. If not, I would like to ask Dr. Nick first. Dr. Nick, regarding on the TRL, I think both of you might share something. How can, if let's say the researcher would like to have a kind of material, how that they can get involved in the TRL, such as that you have shared all those information. I mean, how they get to know all the procedure, all the documentation, everything. If let's say that we want to have a research that can develop a new product or something.
Okay, actually, based on my experience, actually, I got to know this TRL last year because what they call this, the MOSTI come up with a grant related to TRL, right? And then we got this sharing session with the key person from the National Nanotechnology Center, Mr. Kamaruzaman, Mr. K. We know that as Mr. K. And then he mentioned about the TRL. So so actually it's a little bit new lah TRL ni as for me, and I do not know because when I find out the TRL ni in related to the materials development and materials or chemistry development, there is no such thing as TRL. TRL ni is for the engineering, especially for the space engineering engineer. So that's why I I propose TRL level, different level based on my experience, and also based on the requirement needed if you want to commercialize the product. If you want to commercialize, because this biomaterials is related to human, right? Fertilizer is different product because we use it on top of the soil, so not related to the human. So for the human, we need to have an NPRA, chemical testing, in vitro, and so on. So we need to put this safety, what they call this, related to the safety at the early stage, which is TRL 4 to 5. If not, because it is actually related to the approval by the NPRA at TRL6. So we need to achieve that before we go to the next step. So that's actually the good thing about TRL ni. TRL ni we have the plan along the way, starting from the basic research up to the commercialization, and we fill in the gaps between the basic research and also the commercialization part, because there's actually a huge gap, and then we need to have a bridge to what do you call this to get from one part TRL4 to the TRL 7, for example. Okay, so TRL ni is based on the what do you call this our own research. That for biomaterials ni, sometimes for biomaterials, they come up with a simulation, right? Simulation with a heap, something like that. Okay, so that's simulation. That simulation actually is TRL 1 or 2, not up to the 4 or 5, because just on the simulation. So TRL 1 or 2, sometimes maybe TRL 1 only because it's hypothesis. Okay, so it's depend on the biomaterials that you want. So that is my question. Okay. Zul
Okay.
want to add?
Yeah, Dr. Zul, anything you would like to add? Anything regarding on the question?
I think Dr. Nick already covered it enough.
All right.
Yeah.
We have one question from the chat box from Dr. Nurhuda Muhammad. There are so many tests available for biomaterial characterization. So how many tests do we need to do to get good findings or outcome? I think both this question might either one of you can give your answer or suggestion.
Dr. Zul, you want to answer this question?
Okay, me first. Actually, for Nurul Huda, I think it depends on what is your subject. Because I'm dealing cosmetics, so we have to look at all the tests that we need in order to get the notification from the NPRA. Okay, because I'm dealing with NPRA in either we have a cosmetics, we have is a food, or it is a medicine, Dr. Nick, kan? So that's a three different things. So if you want to do related on cosmetics, okay, so they don't want, we have to identify, like me, I'm using a sunscreen or UV filters. That's why I show just now the what called the Annex 7. Okay, I mentioned about the UV filters that available. Okay, if not, I have to come out, I have to declare this, what is the new kind of what I call biomaterials or UV filters that I want to put in my cosmetics. Okay, so it depends on what subject you are looking at. Either you want to do cosmetics, you want to do a food related, or you want to do about medicines. Okay, so I repeat, because mine is about sunscreen, or I have to look at X7 because it refers to the UV filters. Okay, so I have to do all the tests related to UV filters, such as SPF. Um, we have another test we call the eye irritation test. Is it what I call hazardous of the what I call logam berat, heavy metals. So that's a very simple, and it's also cytotoxicity test. So that's a very four kind of compulsory test for me to declare as a cosmetic, in order for me to get the notification numbers from the NPRA. You, Dr. Nick.
Okay.
Dr. Zul, is it the same thing for material or for bone graft, for example, that Dr. Nurhuda asked? Is it for example material for bone graft? Maybe Dr. Nick or Dr. Zul.
I think Dr. Nick, because it's related to a bit bone graft related to medical devices, and Dr. Nick, right?
Yep.
Do you have any comment, Dr. Nick?
Yep. Actually, I have one project on this. We come up with hydroxyapatite incorporated silica aerogel for bone implant,
Yes.
as a bone implant. Okay. So in chemistry point of view, first thing when you synthesize it, you need to do a lot of you need to characterize it using several high-end instrument. Okay, the most important is SRD, X-ray diffraction. After that, if you want to support the result from XRD, you need to have a IR infrared spectroscopy to know the bonding. And then after that, you go, you analyze the morphology of this product, okay? Morphology of the material using PSEM or TM, okay? And then you can see the morphology, and then also you can see the porosity. And then you can use what do you call this BET analysis for the to determine the surface area and porosity. Because for the bonograph, the porosity is also very important because there is the porosity is very important in order for the cells to grow inside the bone. Okay, and it's depend on the bone. It's depend on the material that you use. Sometimes we need to determine the what do you call this the surface area. Okay, using argon gas, not using nitrogen gas. So it's depend on that. Okay, so but for me, a typical biomaterial characterization is SRD, infrared spectroscopy, PSEM, EDX, and TM. That is for the main. The rest is physico-chemical properties based on that material. And then after that is in vitro study for the against osteoblast cell lah. Tu memang is very important lah against osteoblast. If you want to use it for bone graft, do not just only focus on fibroblast cell. Focus on the osteoblast cell. Okay, because osteoblast cell is the cell for bone. For fibroblast cells is for the cytotoxicity is okay. Okay, so that is actually my answer.
All right, Dr. Nohda, I hope it answer your question. So I have one question for Dr. Zul. For the bone finch, is it there's a specific criteria the fish type that you use, or is there any bone from the fish that they use for your product?
Thank you, Dr. Fatihah. Actually, we have already looked through the types of bone. I'm looking for the very easy resources because tamban fish, we always doing tamban fish for keropok. Okay, so I know that we able to get the resources in bulk numbers. Okay, back to your questions. I haven't tried. I never compare with the marine fish and also the freshwater fish yet. Okay, but it's very interesting to know when I compare with the chicken and also the cattle bone. Okay, so it's very interesting because the values of calcium is different, but the values of phosphate is higher in the marine fish. Okay, so that's what I'm looking for. That's what I'm choose the tamban fish, even though it's very small, right? But have kind of a high value of phosphate rather than the calcium itself. Okay, okay. Back to you, Doctor.
Dr. Zul. So is there any more from the audience questions? All right, I think Dr. Nick is sharing the answer for the Dr. Nohda. Dr. Zul and Dr. Nick, so we will share the video recorded in Telegram group for 6 months. So in that group, we will the participants can ask a question and we will share with you. Is there anything that they would like to ask? So today I think is the last session for the series 5, and I really feel grateful for both of you to accept my invitation. Inshallah, for the next year we will have a next series of webinar with all of you to have the collaboration with the very tremendous researcher from a very great university from UMT and UTM. All right. So before we end this session, I would like to invite Dr. Nohayati Yusof, the heads for the all series. So today maybe Dr. Nohayati Yusof would like to say one or two to the speakers, to all the speakers that attend for this last session. Please, Dr. Nohayati Yusof.
All right. So, okay, thank you very much, Madam Chairperson, Dr. Fatiha. Okay, she is also the head of Series 5. Okay, much appreciation I would like to send to our honorable speakers for the day, Associate Professor Dr. Ni Ahmad Nizam, and also Associate Professor Dr. Zul Helmi, right? Okay. Okay. As well as our invited guest speaker, Dr. Mohd Harry and Dr. Juan, all the way from UTM joining us. We are, we are everywhere from around the world. So we also have professors, lecturers, researchers, postgraduates, undergraduate students, technicians, and also some of the, people who want to learn. Okay. Today is the last session, so I would like to inform today is the last session in Series 5, which also signifies the last series of all 5, which started in April. Okay, the last series in our online certificate course, which begin in April, online certificate course in lab techniques for molecular biology and also biomaterial development. So thank you very much. Today, I would like, I am here today to represent my team from Series 1 to Series 5, me, including Dr. Wan Nazatushima from Series 2, Dr. Karul Baria from Series 3, Dr. Mugana from Series 4, and also Dr. Fatia from Series 5, who have been with us, all of you, from April till now. And I would also like to thank the supporting members, Dr. Anshid Noor and the team, Usains, who handle all the registration, all the fee, and also who have been attentively respond to participants' requests, no matter what time, 12 o'clock, 1 a.m., they still respond to the participants' requests. And also thank you very much to all of you, our participants, for your kind cooperation, for staying with us all this time for 5 months. You have not missed any of this series for 5 months. Imagine every single week have been staying with us throughout the both Eid, and also we are very happy. We are very honored. I feel honored to be for you participants to join us, and I hope that we can assist you along the way for your research work. And stay in the support group, in the Telegram group. Do ask us, feel free to ask us any kind of question if you do have, and also if you do have any questions to the speakers. And last but not least, I wish you all the best for your research journey. Yeah, and thank you very much, Dr. Fatiha, for for giving us a really good session for today. And I hope all of you, can you please give a big applause to our speakers today by sending us that. Okay, and maybe we can have a Wi-Fi together. Can you please switch on the webcam? Yeah, okay, so that we can have a Wi-Fi together, all of you. Yeah, we can wait for 2 minutes for you to switch on the webcam. Yeah, we can have a photo session, the pandemic session.
The participant, can you on the webcam?
Maybe they can turn on. Dr. Juan, tak payah bermalu ya, Dr. Juan.
Saya kejuan ada.
Otherwise, I will call out your name one by one until you talk. You see, ada yang dah lari. They don't want to turn on the camera. It's okay. Maybe we should just screenshot while there are still people in the room.
Dia kena buat promotion. Siapa buka video dapat produk Zul.
Siapa buka video dapat produk. Kita sponsor.
Diskaun ke diskaun? Boleh?
Aduh,
Okay, 1, 2, 3. Smile. Okay, 1, 2, 3, smile. All right. Thank you very much. Thank you, Dr. Nick. Thank you very much. So inshallah,
Thank you,
call back for the collaboration. All right. Thank you.
Doctor, thank you. Baby, thank you.
All right.
Bye.
Assalamualaikum.
All the best. Thank you.
Salam.